So yesterday we had a 1.5 hours our conversation with a few people about grad school applications, about internships, about research, finding research assistant jobs in labs, and many other stuff. And it's recorded, and you can find here in this link. And we will try to schedule another one in the next weeks with Suki and Michelle. This time it was only with Chelsea, Caroline, and May. So what else? Nothing. Let's begin. So today we are going to go through a short review of the important points that we talked so now, and then we'll talk about receptive fields, cell specialization, and lateral inhibition, which is a very important topic. And next week, we are going to talk about grandmother cells, among many other things. All right, so we talked about light, which are electromagnetic waves that we interpret inside our system that leads to our perception, which is only a very specific indirect interpretation of the physical world that reaches us. So we said that from the white light that comes from the sun or other sources of light that are white, sunlight encompasses all the infinite different wavelengths together, and we, with our eyes, different animals have different ranges, we see only wavelength between 400 and 700 nanometers of wavelength. They, when they reach our cones and rods, this energy with this specific wavelength size, this energy makes a structural chemical change on those receptors that lead to electrical discharges, meaning the receptors as any neuron fires, and the story goes on. Now, I told you last time that different tissues, different materials, they react differently to each wavelength. So very big wavelengths, they don't do anything to most of our organic material tissue. Very small wavelengths they do, because they're very small. They enter through the tissue, and they may, depending on the wavelength, they may interact with the atoms, with the molecules of different tissues. So for instance, X-rays, they bounce on a certain density of tissue, like bones, but not others. They are absorbed by some tissue, they bounce other tissue, or they go through other tissue. So because they bounce on certain density, like bones, we can see through X-ray prints, but they are also harmful, because this energy, when it interacts with tissue, above a certain amount of interaction, it starts to change it. And some changes are pretty harmful, and They can be fast, they can be slower, they depend on accumulation. Microwaves, for instance, which are larger, between 10 and I think 30 millimeters, pretty big. They, on most organic tissue, this energy is discharged and they heat. That's why microwaves, they heat food. This heat can be harmful, for instance, to what we know for proteins. When they are heated, when they get hot above a certain point, their structure changes drastically. Eggs, we can see when when you heat the white of the eggs, which is mostly protein, a little bit of heat and becomes white and it becomes hard. That's why, for instance, high fever is dangerous. High fever above 107 Fahrenheit or 42 Celsius, it starts to change the the proteins of our brain. It's like our brain becomes boiled egg. So, so people die from above a certain above 108 tissue. The tissue damage starts to be really harmful for children, for babies. Even lower temperatures start to change the brain. Okay, we talked about neurons that are very simple devices. So all the neurons have the same anatomy. So we have the soma, the cell body, dendrites for most cells inside the bodies, or for all our receptors in all senses. Instead of dendrites, they have receptor devices that react to some kind of energy. It can be chemical, if it's smell and taste. It can be light. It can be to movement also for hearing, etc. Communication is either electrical or chemical electrical within the cells, or chemical among them, between neurons, which occur by mostly diffusion and magnetic attraction or repulsion, which are very simplistic processes and slow. And also not dedicated, like it's things that randomly happen in a certain area. So it's not a very effective and fast dedicated mode of change. And for every change, for every firing, every neuron has to recharge, the reuptake. Different neurons, different times, different inner mechanisms with the neurons. But all of them depend on this refractory time. Intersynaptic communication, the chemical communication between neurons, can be excitatory or inhibitory. So depending on the neurotransmitters released or many other mechanisms, it can be indirectly via reuptake, which enzymes that dissolve those neurotransmitters swimming randomly around the intersynaptic cleft. So all in all, they can be divided in either excitatory or inhibitory. All right. In some neurons, all of them, they are very simplistic devices. They work unidirectionally from soma to axon. They can be, they either fire or not fire, and they can release either excitatory or inhibitory neurotransmitters towards the next neuron. And they all depend on the time to reuptake, and the difference of any event is in their firing rate. The frequency that they fire when nothing is happening, they have a baseline resting state, different neurons, different frequencies, and then when something happens, either this resting state baseline firing rate decreases or increases. Rods and cones, many difference between them. So the kind of stimulus they react to, so rods react to these around 500 nanometers wavelength. It's not that restricted range. As you can see here, it's a gradual range. So wavelength around between 500 and 550, they actually they activate rods, green cones and red cones, but not blue cones. Maybe even if they short a little bit of the red cones. So for every specific wavelength that reaches our retina, all the cones to a certain degree will react, but they will react more or less strongly depending on their specific sensibility. We'll come back to that. This is very important for us to understand color perception that we'll see next week. Reuptake duration. So we said that cones, they recharge, they reuptake much faster than rods. And that's the difference that determines the time of dark adaptation. Cones, they reuptake much faster, but they reach a less sensitive point. The maximum sensitivity is less than rods that reach their maximum sensitivity much later. The difference between, let's say that you are under normal conditions, light, and then you enter, the lights are turned off. After 25, 30 minutes, you reach maximum dark adaptation for both cones and rods. At that point, all in all, your sensitivity to light will be something like 100,000 times more than when you were adapted to normal light conditions. So we have a huge range of adaptability between light and darkness. Another thing that I remember later, I told you that in the army, when soldiers prepare for a mission in a dark situation, they cover one eye beginning half an hour before in order to have at least one eye adapted when you arrive to the soldiers have to arrive in the dark. So I remember later that I read somewhere that there is a hypothesis that that's the reason why pirates also used I don't know, we don't have pirates, used to use eye patches. I don't know, at least me as a child, I always thought that because they are all the time, poking everybody, so most of them lost one eye on the way, which, if you think about it, doesn't make sense. That why pirates specifically lose one eye all the time. So no, it's because they, for the same reason that soldiers patch, because in those big ships, in the galleys, that's the way to say, they all the time go down under the deck where they didn't have electricity. It was dark, or much more than, and on the deck, sun. So they had all the time one eye covered. For anytime they go under the deck, they open the patch so they can see. And when they go up, they work with one eye. And if they have to be more, I don't know, if they needed better performance above the deck, they will take it out. It's not because they had one eye. Okay. Distribution. So we have much more rods, and they are not in the center of our retina, not in the fovea and hardly in the parafovea. And our cones are mostly in the center of our retina, where we don't, in the fovea itself, we don't have rods at all. And cones we do have everywhere, and they are evenly distributed once you are outside the parafovea. This is 10 degrees, so you have 20 degrees just for you to understand the size of this area in terms of your visual field. So 20 degrees for arm length would be something like 2.5 feet. Would be something like this. Can you see my would be like 3 pounds in front of you at arm's length. That would be 20 degrees of visual field. So from, you can see me, right? So from this distance at arm's length, from here, we hardly have cones. But here are most of the cones. All right. Convergence, this is important. So convergence means how many receptors converge into one ganglion cell. They differ, rods and cones. So many rods converge into one ganglion cell, and cones in the center, in the best case scenario, in the center of the fovea, they have 1-to-1. It's not even convergence. One cone is connected to one ganglion cell, which means that rods are much more sensitive to light, because you have, if a ganglion cell needs 10 units of activation, so the same amount of light falling in the same area will activate that ganglion cell. And here, if it's 1 by 1, you need 5 times more light, or an area much, no, 5 times more intensity to activate one ganglion cell in the fovea. On the other hand, because of that same relationship, our cones have much lower resolution than rods, right? This is clear, right? Stop me if... We also talked about the organization between the area in our retina after the optic chiasma. All the information from the same side, the right side of both retinas, they end up in the right side of our occipital lobe, in our extriate cortex, responsible for vision, and from the left side to the left side. So it's ipsilateral. Our visual system is roughly defined as divided into the dorsal stream once it arrives from the eye to the occipital lobe. It's divided into a fast but not very with low resolution, let's say, the dorsal stream where things happen, movement, and the ventral stream where it's associated with much more information, memory, object recognition, semantic, everything that we can say the what identifications. And this spatial, the spatial organization that we have in our retina is transferred to our visual cortex. So adjacent areas in our retina, they will reach adjacent areas in our cortex. I'll explain it soon. Where were we? Receptive fields, very important. The idea is the same for all our senses. A receptive field of any given neuron is the geographical, the spatial area in the receptive, in the area of its of the receptors that send information to it that it is sensitive to. So it can be a neuron in somewhere here that reacts to everything that happens in a specific area in my skin. So for for the ganglion in the visual field, so let's say this ganglion cell will be affected, its firing rate will be affected by anything that happens in this specific area in the retina. This other one, it's this area. So the further away from the retina, the larger the receptive field, and receptive fields, they have shared area, they interthink. So as I just said, the further away from the retina, the larger the receptive field. In V1, V4, which is the extra, it's these little areas here. We still have very small receptive fields, so high resolution, but in in the temporal lobe, this area in the end of the dorsal stream, where, for instance, objects and semantic meaning begin to be attached to visual stimuli, one neuron reacts to a receptive field 25 degrees, is something like 3 feet from in arm's length. So one neuron reacts to most of the central visual field. They obviously have a lot of shared area. Cell specialization in the visual field. Okay, I will show now drawings of animals. I just let you know, it's nothing horrible, it's only drawings, but some people are very sensitive to see animals in a experimental setting, so I'm giving you a moment to look away. It's only 3 slides, okay? Anyone that does have, please write. Well, I'll do it anyway for the people that are not here and watching from home. Single cell recordings, that's how most of the information that we have about cells in our visual system we learn from. We'll see other stuff later, mostly from the 60s, 50s, 60s. Single cell recordings work like this. You put the animal clamped into a fixture. They are under anesthesia, of course, but it's not, it's a specific, it's not full anesthesia. So their eyes, for this kind of research, of experiments that we'll see, their eyes don't move, but they are open and they react. They are not, well, I don't know about suffering. I suppose it's assumed they are not feeling pain, but how much not pain can you feel? I don't know. Anyways. And then another fixture is attached to their skull in a specific area, within which a very, very thin electrode, well, I don't know how, it's almost invisible, and it's inserted into the brain, into the cortex, until it reaches one cell. Which cell is chosen? It's not chosen. Whatever cell it arrives, it works. It's a kind of trial and error process, because the probe, when it may touch two cells at the same time, it may touch a small dendrite, it may touch an area that, so sometimes it takes many trial and errors until you receive a signal from one cell that sounds stable. Sounds because the what from this probe, it's attached to an electro device that every time that there is a discharge, it tuck, 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 tuck. You can also see in a screen. Nowadays, we have high resolution information about the tomography, about the anatomy of each animal you want to probe. With magnets, you have 3D tomography. But in those days, they had not such a good way to know where they, so they knew more or less where they want to put it, but so it was not a simple procedure. So this, so it usually looks like this. This is for a more behavioral setting where this monkey is not under anesthesia, local for sure, local anesthesia in the skull, but this monkey is conscious. There are many tasks that monkeys learn in the field of attention. There is a lot of research based on monkeys, because only on monkeys they can learn complex enough tasks, and we can I don't know, science feels that we can put things inside their brains. Well, and they learn through the rewarding, the way that they learn how to do what to do is that they receive drops of juice into their mouths every time they do what the researcher wants them to do. So the following experiments were based on cats. They were not responding to it, they were not learning anything, they were fully anesthetized, but their eyes were working, like, and they had a probe in their ganglion cells. Okay, so out of so let's say we in those experiments the researchers probed a ganglion cell that they assumed was receiving information from a certain area. They had an estimation of the receptive field of that specific ganglion cell. So they started to put light on that area of the visual field that the cat is looking at. This is the baseline resting state of that ganglion cell. So every feels, it sounds like tick, 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 meaning nothing is falling on that receptive field. And then they showed a small spotlight in the center of that area. Nothing happens. They enlarged in the same space, they enlarged the spotlight. Suddenly they hear the ganglion cell taka 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 taka, meaning okay, first of all, it is the right area, it is the right receptive field. They continue to enlarge the spotlight. Suddenly the ganglion cell starts to react slower than its resting state, so it's being inhibited. Larger and even more inhibition. Anyone wants to try to guess what's going on? No, no one. Okay, so I'll tell you. So these ganglion cells, and I don't know if all, but most ganglion cells, they have quite complex reactions to light within their receptive field. So the entire thing is the receptive field, but they react differently depending how the light falls. So in this example, in the center, they react by increasing the firing rate, but if the light falls on the outside area, not in the center of the receptive field, it inhibits their reaction. Light that falls only in the outer circumference, they have only inhibitory effects on those specific ganglion cells. So this is from the 50s. I think it was the first big publication about those attributes of ganglion cells from 53. This is from Wiesel, 59, very important work. I'm jumping. So you understand what you're seeing. When it's small, this area, the ganglion cells start to fire like crazy. When the light is outside, it stops. And when everything is lighted, it's something in between. So we have many different kinds of ganglion cells. Center on, center off are the two main. So this is the different situations when light falls in the central area a little bit, outside a little bit, all the central area, maximum firing rate, all the surround, maximum inhibition, okay? And the opposite, this is the center off, where the exact opposite happens. As I said before, the same kind of organizations happen in all our senses. No, not all our senses. Skin, yes. Now I'm not sure regarding other stuff. I think it is. And if not, so these kind of specializations, they exist for all our senses. I don't know regarding specific off-center, on-center. David Hubel from Canada, Thorsten Wiesel, who is still alive, he's 19 years old, or 96 years old now, he's from Sweden. They received for this kind of work a Nobel Prize in 81. Again, I'm going to show you a cat, a drawing. That's what they did. They probed the visual cortex, this area, the V1 area, in an anesthetized cat, and they showed bar lights in different orientations moving around the receptive field of that single neuron in the V1 area, in the visual cortex. This is the original recording of that experiment. So what you see here, if you see, the noise is the, can you hear? Yes, okay. So the noise is the cell firing. So is the resting state. When light falls in the receptive, in the visual, in the receptive field, it increases or decreases depending on the specific way it falls. It can be filling the entire receptive field, and as you'll see soon, it depends on the orientation. I'll continue. And then they mark not firing, outside receptive field, nothing. A little bit, resting state. Oh no, sorry. As you can see, when a lot of light, when a lot of light falls outside the receptive field or beyond, it completely shuts the neuron. It's less than its resting state. Let me correct what I said. Where he marked the circles, I said it's outside the receptive field. It's not. It's inside, but this is also an on-off neuron. So it not only is sensitive to the orientation to a line. If you put another form, it won't fire as well. And within the receptive field, this is where the circles denote the inhibitory, the off part. Here too. It literally shuts the neuron. Now the same stimulus, but a slightly different orientation, and nothing happens. Nothing. Okay, it's clear what happened, right? So it's impressive. I don't know, I find it impressive that here in the back, there are neurons, one single neuron, that the only thing it cares is about a specific pattern in a specific orientation in a specific area. This is from the original paper. So these are the spikes according to orientation, size, width, etc. And they found out, all in all, that our visual cortex, as I said before, it's the organization of the cells is follows the spatial organization of their receptive fields. So if you move from one cell to its neighbor, it will react very similarly to its neighbor, but a nanometer, no less, a decimeter to the site on the retina. If you move radially, if you move on this direction, so the changes are in orientation. If you move in another direction, the change can be on width. So it's a very highly specialized distribution of cells. In other cells, in our in view 1, this is still view 1, the center of the striate cortex, we can find, they found cells that react exactly the same, but depending on the area around it. So look, this cell here, it's the same one cell that reacts to this object with this orientation, but it will only react when it is within a larger area, even outside its receptive field, that has other objects that have the same orientation, because it when the area with the same orientation is different from the larger area around it. So look here within the receptive field, or maybe outside it, and here is exactly the same. But this is an object that perceptually is different from the background, and then it fires. And if it's not different, it won't fire. This is another kind of specialization, the same idea as before. So the same stimulus falls in the same area, the same receptive field. The larger background here, nothing happens. But when you have one set of objects that follow that has some kind of meaning in terms of similarity or continuation, then it fires. Okay, how much time do we have? Not much. I will jump. No, I'll say it. So again, we are learning more and more about attributes and activity of our neuronal system. But perception is a translation, it's an indirect interpretation of what happens. Information comes for a overall interpretation of all this endless activity a long time, and that ends up in our conscious or even unconscious perception of what happens around. So it's mediated by a lot of different inner interpretations of simplistic processes. One example, this is it's a Bloch's law. It's a very simple concept that explains a lot of things And for some reason, it fails to appear in a lot of places that it should appear. I don't know why people, I don't know, it happens all the time, that people forget Bloch's law. Bloch's law says like this: our perception is identical if within 100, it's not written here, but within 100 milliseconds, it's not, it's a short period of time, but it's big enough perception wise in terms of processes that go in our brain. It's a long window of time. So whatever happens within 100 milliseconds in terms of change in luminance, in terms of the amount of photons that reach our retina, our perception will be identical. So if the same amount of energy of light is here, it's represented by the area of these graphs under these functions. So perception-wise, this is identical to this, which is identical to this. So a short burst of light, is the same perception-wise for a third of the intensity spread along 100 milliseconds. So a burst of 33 milliseconds is the same like less intensive 100 milliseconds. And it can be also, this is called summation or integration. It's the same idea that even three pieces of information that, again, inside our brain, inside the visual pathway, there is a big difference. Though the neurons here, they know the difference between this and this, but our sensation, our perception cannot discern. We have 25, no, 50, we have 20 minutes, right? Okay, lateral inhibition, that's very important. The idea of lateral inhibition is the amplification of a given perception by shutting down, by inhibiting everything around it, by inhibiting the background and intensifying the foreground. It would be like augmenting the contrast in a picture. So usually in the textbook, it's usually defined in the neuronal spatial-based explanation, but it's much more than that. First of all, it happens in all our senses. It's an expression of perception. It happens all along, in different ways, along the process, the pathway of perception. It can be based on spatial organization, but it can be also not. We'll see soon. Can you see the strange thing that is occurring here? Can you see that whenever you look, let me show what you have. I cannot put it. So in the crossroads of each one of these points, you have a white circle. But wherever you look, you perceive that the other circles away from the center, wherever you fix your gaze, they become darker. Can you see it? Yes, is it working? All right. The same here. This is the Mach Bands. Do you perceive that this area feels darker than the rest? A little bit, and this a little bit lighter than this area. Does it feel like a gradient here? Can you see through on the? Okay. What happens is that between neurons there is those inhibitory interneurons. In the case of our visual system, those would be these little guys that mediate between our receptors and the ganglion cell, the amacrine, the bipolar, and the horizontal. Not all of them are completely understood. This is ongoing stuff. I think the amacrine cells are not fully understood. Maybe I'm wrong, but they are responsible for that in this specific, in the case of our visual system. So whenever a given receptor is activated, it not only activates its private ganglion cell, it also inhibits the neighbors. So let's say that this is a, well, looking at this diagram, it would be more like a bipolar cell, not a ganglion cell. So, but it doesn't matter. So it's a cell in the second or the third tier of layers after the receptor. So the maximum activation of this cell would be if light falls on one of these three receptors. Most of all, if it falls on all these three receptors. But as we can see here, that if it falls only on 4, we have a certain level of increased firing rate, which is identical to the firing rate if light falls on 2 and 6, which are outside. These are receptors outside the receptive field of that cell. So light in the neighbors, 2 units of light in the neighbors is the same perceptually and already in the second or third layer of the visual pathway is identical to 1 unit of light following the center of the receptive field. And look what happens if, well, it follows between 3 and 4. This is the maximum. And if it falls a little bit further away on 1 and 7, suddenly we have inhibition. Okay? If it falls even further away, it will not affect it anymore.
Just a quick question.
Yes. Yes.
So in the graph, does the 2, does it mean 2 to 6 or 2 and 6?
2 and 6. 2 and 6. If it was 2 to 6, it would be larger, but it wouldn't be as large as 3, 4, and 5. So 3, 4, and 5 would be the maximum fire. Here it's only 3 and 5 without 4. It's only the stimulation of two receptors. So this would be the maximum, I don't know, probably here. And then if you include one neighboring one, it will go a little bit less, and 2 neighboring a little bit less. So the maximum inhibition would be when this guy doesn't have any light within its receptive field, but its neighbors are being activated. Okay.
Okay.
Here, can you perceive the darker shade here? Okay, it's exactly the same. It's only a perceptual distortion. This is the actual, well, sorry, I'm showing you the wrong thing. Here, these are exactly the same. There is no gradient. It's called the Mach band. It's the same Mach of the Mach number when that in speed of sound. You so the speed of sound, because is counted as in numbers of it. So an airplane, we hear when there is acceleration beyond the speed, oh, we hear Mach 1, 2, 3, 4, 5. So it's the same guy, and Ernst Mach that decided. So this is the Mach band. What happens is that, so this is the light intensity, how it falls. So this area A here is identical to this area, and this area is identical to this area, but what we perceive is this. How it happens? This guy, this neuron here, is being equally inhibited by its neighbors that are receiving the same amount of light. So the inhibition, the lateral inhibition that this guy is receiving is, let's say, -10 and -10 from both sides. But this guy, while it's receiving -10 from this guy, it is receiving, because here less light is falling on this receptor. So less activation, so less lateral inhibition. So this guy is inhibiting this guy less than this guy is being inhibited by this and this. You see the numbers here, right? It's clear. So what happens is that this guy has less inhibition than this guy. So we perceive more light, as if it was more intensively activated. This guy is being inhibited less than this guy because of this neighbor that is inhibiting 10. And this has the same from both sides. The same thing occurs even from non-adjacent areas. The illusion here, the perception is that this is darker than this, right? Everybody perceives that. Yes? Okay. But they are not. They are exactly the same shade. What's happening is lateral inhibition because of the foreground of this gray area. Okay? The Hermann Grid, which is the first thing that I showed here. So this is, I put it here, it's not exactly the Hermann Grid, it's only, the illusion is stronger. The Hermann Grid are only adjacent dark squares. And what you are supposed to perceive, I don't know if through the, whenever you look at one of the crossings, do you see, don't move your eye. Look here. Do you perceive a darker shade on the other crossings? Yes? Good. And a little, so the darkest shadings are here, are on the other crossings, not in the center, but also we also perceive an intermediate darker shade in the corridors, right? So it happens because these guys here, they are being laterally inhibited by the largest numbers of neighbors with the largest amount of light. So in this case, from 4 highly activated neighbors. Okay, so this is the, and if this neuron was separated from its neighbors, it would be activated by 100 units. But because it has 4 inhibiting neighbors, the perception is much less. And those guys in the corridor, they are being inhibited only by 2 neighbors. So that's why we perceive a lesser shade of dark in the corridors. Okay, so as I said before, lateral inhibition is not only spatially based, and it's not as simplistic as usually it's defined by neurons in the visual cortex. For instance, the white's illusion. Do you all perceive this as much darker than this? Yes, yes, good. It's a strong illusion, but they are the same shade. Now, if we try to explain, if we try to understand it according to lateral inhibition, it doesn't make sense. Because look, these that we see darker, they are actually being much less inhibited than these guys. They are inhibited by these highly activated neighbors, but they are hardly inhibited by most of their neighbors. This, on the other hand, should be highly inhibited by all those highly activated neighbors, right? So what happens here, sorry. So what happens here is that well, this is an explanation suggested by Alan Gilchrist, I don't know, in 1999. And it's the idea of belongingness, meaning that we perceive these lines, these gray lines, as one object that is being occluded by these black lines. You see it, right? You feel it, that it's one object occluded by these lines, meaning that this rectangle belongs to a white background. This, which is physically wise in terms of light, is identical to this. These guys we perceive as one object, one rectangle that its background or its belonging is to the black lines, right? So our system is perceiving gray on black in this case and gray on white in this case. So it is lateral inhibition, but it's not in the initial stages of the visual pathway, is in a much higher, probably later in the dorsal pathway system, that starts to organize this entire picture into something meaningful, meaning there are objects, there are entities, there are dimensions, There are relationships between these three objects or two objects, each one belonging to a different area. So these are interpretations that do not exist here. Much higher, much later, and that lateral inhibition is much stronger than the neuronal lateral inhibition that should have rendered the opposite effect. What you perceive the opposite? You perceive this lighter? I cannot hear. Try to...
This, exactly as predicted. I perceive it darker. I'm just saying that it appears though that it's in back of dark lines, horizontal lines that would be in front of it.
So you see this as one object that is darker than this?
Yeah.
Ah, so you are okay, Charles. Nothing wrong with your brain.
Right. No, but what I was saying is the perception though is that it's kind of behind bars, behind horizontal bars.
Yes, it's being occluded, that it's one object occluded by these bars.
Yeah. Okay.
And this object belongs, I don't know, is on a different background. It's in the back, and the back is white. That's the belongingness of this idea. Okay? We have 1 minute, and it's exactly where I wanted. The last kind of non-spatial lateral inhibition would be color perception, that we'll talk next week. So color perception, just to throw it, there are two theories. They can explain a lot of things, each one by itself. They cannot explain everything, each one by itself. I think, and I'm guessing, that probably most of the literature assumes that these are two systems that work together. So we have the trichromatic is the simple one. We have our different kinds of receptors. These are the cones. The rods don't see color, right? They do, but let's put it aside. So a combination of how each one of them for each wavelength, they result in our perception of color. The other one is that we have pairs of two colors. I'll explain next time, that we see colors by the activation of each one of these pairs, inhibiting the opposite perception that is not presented at all, that doesn't exist in the real world energy that is reaching our retina. Okay. Also, next week, I am going to tell you that apparently there is one cell in your brain that reacts to everything that is your grandmother, your specific grandmother. It can be her face, it can be her face like this, it can be her voice, it can be even you remembering, thinking about your grandmother, and then that cell will fire like crazy, but only to that. So it appears. We'll talk about it next time. Okay, we are done. Any questions? Yes.
Okay, I put in the chat a link to the paper that the basis of our Gaddafi 4, just because the conclusion was, and I put the relevant from the abstract that the Chevreul illusion wasn't explanatory completely for lateral inhibition.
The opposite, that lateral inhibition cannot explain the Chevreul illusion.
Sorry, yes. So, because we were looking at that, right?
Good.
Okay.
So it made sense what you learned today?
What I'm saying is it's partly in conflict of what we were discussing, no?
Why?
Because the lateral inhibition is not sufficient.
Yes, just like we just saw here. Lateral inhibition, it's not only not sufficient, it's the opposite.
Right, okay.
Lateral inhibition is a family of, that's why I presented it initially as something like this. The background enhanced on the account of the foreground enhanced on the account of the background, a polarization. And it can be within a smaller, very technical, spatial-based area, or it can be in larger conceptual processes, color, for instance, and we'll see. And this is only one mechanism that is present in any piece of perception. I don't know if any, but it's everywhere. It's all the time. Sometimes it is congruent with other mechanisms. Sometimes it goes against, and you have synthesis to any direction. Okay. Anyone else? Yes.
I wanted to ask if, I mean, I'm not sure how much this can really be like discussed in literature, but like evolutionary wise, is there a reason that like, because I noticed that the curve for the colors that rods can perceive is more in like the blues and greens that we're very like surrounded by in nature and such. So is that why rods, yeah, so why rods that we have more of would be more attuned to those colors?
Why it is like this?
Yeah.
It's the same like asking why we have two legs and not wings.
Yeah.
This is what happened. We can try to. I find it hard to even say that this is the best solution for anything, since we don't have nothing to compare. And actually, all the animals have better stuff, better apparatuses than we have. They see more light, they hear better, they smell better, they do everything better than we do. No, not all of them, but it's what happened to us. We can, in hindsight, I think that all we can do is explain how things happen and connect to events and be able to predict things. But why? I'm being annoying with you.
It's okay. I mean, I thought it might have been sort of a stretch because I was just thinking about like environmentally, like blue sky, blue water, green grass, like that.
Let me try to be less annoying with you. I suppose that we could say that most of the relevant textures that we mammals, humans, need to discern for food or danger are within this wavelength. Meaning that during evolution, whenever, and it happened, I mean, I don't know if it happened, but it makes sense that maybe, let's say that it happened, that some humans suddenly they had receptors that were sensitive to infrared. They had no, there was no advantage. And if those receptors took the place of those receptors that perceive red, that are part of important things that we have to perceive, then those individuals, those mutated individuals that could see infrared, they had a disadvantage and they didn't continue. What crossed your mind? I'm curious. What when you asked, what did you started to say it, and I interrupted you. So what was the connection that you started to make?
Well, I was thinking about, like I explained before, with like natural colors, and then because I was also thinking about how we have so many more rods than cones, and I thought that maybe there might have been a potential relationship between the number of rods in relation to environmental colors and surroundings in a sort of like historical, like now obviously we have the ability to manipulate like pigments and such, but ancestor wise, when it was settling by fresh water and healthy fields and etc. It's all just speculation. I was
No.
you know.
Let's play it now. Speculation is interesting.
I probably have to go in a minute. Sorry to interrupt, but because there's my recitation comes up. But there is evolutionary theories about why we have color vision, because a lot of mammals or most mammals don't. And so it would be related probably, I think, from what I read, to food supply, being able to judge when, say, a fruit was ripe or something like that. So that doesn't answer your question, because you're asking why rods are predominantly sensitive to the frequency, and I'm answering a different question, which is why we even have color to begin with. But thanks.
Thank you, Charles.
Right. See you later.
See you later. I'm coming to Michelle in 1 hour, so I'll see you there.